Hello and welcome to video 3 of the WestGrid Computing Summer School Bioinformatics session held on June 17th, 2020. So far today we've had an introduction to next generation sequencing data and diverse data types, transcriptional cis-regulatory data sets and where to find them, and then in video 1 we initialized our workshop directory and began to explore data. In video 2, we mapped and converted this data using BWMM and SAM tools and generated bigwigs with deep tools. And now in video 3, we are going to visualize the data using IGV. Now in this video, we are going to be transferring files to our own computer or using a mount point like SSHFS. And because of this, I recommend starting this transfer early and then pausing the video and continuing. We also will download and open IGV or Integrative Genomics Viewer. We will load that data in IGV. And then lastly, we will zoom into a region and take a snapshot. So at the end of our analysis pipeline, we have a sorted.bam file and a associated index.bai file. And we're going to use those to visualize in IGV alongside the bigwig or the .bw file that we also created. So transferring data from the server down onto your local machine, there are several ways you can do this. Just make sure that you grab the sorted bam, sortedbam.bai, and bw or bigwig files. Bring them onto your local machine. You can use FileZilla, which is shown in this window here, available at this link. And with this information, the host, username, password, and port 22. Alternatively, what I'm going to use is SSHFS, and this allows you to mount, if you have a Mac, it allows you to mount the server onto your own computer so that you can access the files on the Cedar compute cluster remotely. Information about how to get started with that can be found here. I don't know the equivalent example for PCs or Linux boxes. While you're transferring, make sure that you download IGV. So if you don't have it already installed or if you want the latest version, you can go to this website and download it for free. I grabbed the IGV Mac app. And then open up IGV. Try that again. Okay. Opening up a new one. We'll get a little bar that looks like that. Loading genome. And you should have a window that looks somewhat similar to this. Now the IGV or Integrative Genomics Viewer, we have the contigs or chromosomes in our reference genome. We have the ability to change reference genomes up here in the top left by clicking on this or zooming into a specific chromosome. And the navigation of this we will also explore a little bit more in the next few minutes. So when you have IGV open, load HG38. So if you don't see HG38, for example, right now I have it loaded by default, but if I didn't see it, you can click more, and you can see these are the genomes that are already in IGV, and if you need assistance in creating a reference genome for your own purposes, perhaps your organism is not listed there, then contact me and I will assist you. So the first thing that we're going to do is we're going to go File, Load from File, and we're going to load the BAM and the bigwig, which we just created. So I'll go File, Load from File. Let's see if this is still mounted. Nope, it is not still mounted. So what I'm going to do is I'm going to show you how I would do the SSHFS using OSX Fuse. So what I will do is I will type SSHFS. I want to mount Richmond P at cedar.computecanada.ca or whatever your username is. And then we are going to want to mount Scratch Richmond P training June 2020. And we're going to set this to a local directory that we have made beforehand. So for me, I'm going to set it to this directory that I've made called Portal 3. Yes, I'll need to enter my password. And now, if we look at what used to be Portal 3, inside we can mirror this directory. So now this is appearing on my local machine. Now coming back to IGV, because I have access to now all of these data files, it's very simple to go File, Load from File. We're going to find that portal. So this is called OSX Volume 2. And I'm going to go ahead and load ATAC-seq bigwig file. I'm also going to grab the BAM file, and I'm going to click Open. So it's going to think the wheel is turning. It may or may not be 10 p.m. the night before I teach. Hooray, grad school. Okay, so now we have our files loaded. We can load a couple more in a minute, or we can zoom in to a specific region in the genome. The one that I've picked beforehand is SPTBN1, which is a gene that we know is expressed in the heart ventricle. SPTBN1. We can type this right into the search box here, and that will zoom to our gene. So the gene track down here, you see this has appeared, and it has the gene SPTBN1. And if we zoom in a little bit more, you can see before we were at 200 kilobases, now we're at 24 kilobases, and we're now able to actually visualize our mapped reads. So up here, this is a coverage histogram for the bigwig file, and this is the underlying mapped data. So we can continue to zoom in here at this 5' UTR of SPTBN1, and as we zoom in, we can see each of our reads, which are actually mapped against the reference genome, as these gray boxes. You notice that these boxes have a directionality to them. You may also notice that several of them seem to have these colors on the individual reads. And this is because these reads likely have some adapter sequence, which we decided not to trim out from our dataset beforehand, although it seems that our reads are still able to align uniquely against the genome. So one of the things that we're going to do here is we can play with a few different options. If you right click or control click there, no, just right click, we can change the track height. Let's make this 100. We can also change the data range. So let's make this 0 to 50. Oh, that was not high enough. Let's just say 0 to 100. And now as we zoom out, you can see that characteristic profile of attack-seq reads around the transcription start site of a given gene. We can also load our other files. So I'm pulling here from Sherlock, but you could also just pull from the files directory if your files haven't finished processing. So we already have attack-seq data. Let's also grab polymerase and H3K27 acetylation. So we're going to grab the bigwig. We already know what the BAM file looks like, so we don't need to grab the BAM file. We're just going to grab the bigwigs for both of these samples. It's going to take a moment to load. Double check on that in a minute. See if there was anything else included that I'm missing. Nope, that looks like about it. Now it's started to appear. Okay. There we go. So, we now have 3 bigwig files. Let's go ahead and get rid of our BAM files so we can highlight things and remove them. And let's make both of these tracks also 100. It looks like the scale on this one is set to a very awkward size. So let's make this also 100 and see how that looks. That looks pretty good. And let's also change the data range here. 100 for the polymerase. Now, so that we don't think these are all the same thing, we can change the track color. So we can make one of these green. And we can make one of these. What's the polymerase color going to be? Let's go ahead and make it purple. That's more of a purple. There we go. Now we can zoom out and you can see some interesting trends about where there is open chromatin or histone marks across the genome. Characteristically, we see these types of marks at the 5' UTR or in the promoter near the transcription start site of genes. Now we can zoom. One of the values of bigwig files is that you can zoom out quite far. So you'll notice here we're at 82 megabases or 82 million base pairs, looking at dozens of genes beneath here. And we can zoom in at some places where maybe we see a discrepancy between these. KIF5C. We can keep zooming in. This looks like it's probably an artifact. If I had to guess, this will be some kind of repeated sequence. Maybe not. Anyways, you can explore your data and you can take some snapshots and save them by going File, Save Image, and then this will pop up. Let's go ahead and save this here. We'll call this Workshop Image 1. Save. And that is it for video 3. There is a problem set 2 where I want you to load and visualize data from at least 2 experiments. If your scripts haven't finished running, make sure that you use the files within this directory. Change the colors of the different tracks, set their height to 100, and take some snapshots, perhaps of a transcription start site. Optional, find a pileup of these signals outside of the genic regions in the genome These will be enhancers. And lastly, email your images to me. Bonus points if you have the capacity to take a snapshot of a data set that you downloaded, which was not one of the ones that I processed, and you were able to map and visualize yourself. Other than that, thank you for attending this session and for participating in these videos. Hopefully this was a helpful format in this awkward coronavirus time. Thank you and good day.